Hi guys, Jamie Batts here, your instructor for Bio 221. This is part 2 of chapter 26, and I'm going to do a little graphic organizer of all of the parts of the nephron for you so we can paraphrase their functions, and then we'll take a closer look at the actual structures of them in terms of histology and so on. So, we have the awkward whiteboard, as always. The nephron will be our middle word. Is the red okay? Yeah, red's good. Okay, so nephron. We talked about the PCT already. The PCT, or proximal convoluted tubule. This is mostly for reabsorption. And when we're talking reabsorption, we're mostly reabsorbing ions like sodium, right? Things like that that your body may need to retain. And it has those microvilli to help with that process. So the next stop on the tour of the nephron, and I'm just going to go counterclockwise like this through the nephron. Doesn't really make sense why I did it like that. I just picked that way to do it. So we have the nephron loop, or the loop of Henle as they are sometimes called. The nephron loop uses something called counter-current exchange or counter-current multiplication counter-current multiplication is what it says, to increase the absorption rates or reabsorption rates within the nephron. So this is where your filtrate gets super concentrated. And there's two parts to the nephron loop. First, there's a descending limb, and there is an ascending limb. The descending limb has a thick section and a thin section, and the ascending limb also has a thick section and a thin section. The thick section is for reabsorbing solutes, solute reabsorption. Solute reabsorb. I'm losing space down there in the little corner. Solute reabsorption on the thick section. The thin section is for water reabsorb. And you might say, okay, well, how does that work? Well, we're going to look at it, and actually I'm going to post a couple YouTube videos. Post some YouTube videos from my YouTube video. How redundant. Post some YouTube videos and textbook videos of the process, because it's actually a really cool process how it works. Basically, as the solutes are being reabsorbed into that peritubular fluid, that changes the osmolarity of the peritubular fluid, which is going to draw out more water. So the reabsorption of one is going to increase the reabsorption of the other. And then as the water dilutes that peritubular solution, it's then going to pull out more solutes from the descending limb. So it helps to multiply, hence countercurrent multiplication, the reabsorption effects. So it's a really interesting process that your kidneys do. And the whole purpose is to super concentrate the urine so that you're not getting rid of the stuff that you might need. So nephron loop came after the proximal convoluted tubule, after the nephron loop, after the ascending limb. That loop comes right back up, and then we're at what's called the DCT, or distal convoluted tubule. Whew, that was a shoulder workout. So the DCT, or distal convoluted tubule, this is for what's called selective reabsorption. This is actually super, I love the kidneys. I think they're super cool. Not only do we have this really cool countercurrent multiplication happening here with the nephron loop, we have what's called selective reabsorption happening in the DCT. And what this is based on hormones. Remember those lovely hormones that we talked about at the end of chapter 18? They came to haunt us a little bit in chapter 21 when we were talking about blood volume and blood pressure, ADH, angiotensin, right? Remember those guys? Well, how did they work? If you remember their mechanisms, what they were doing is they were either increasing or decreasing absorption of water at the kidneys, right? If your blood pressure was very high, then your body would want to get rid of as much water as possible to lower your blood volume, in which case ADH would not be released. And I'll draw all of this out, but I just think it's so cool how it works. Either way, as ADH and angiotensin are present or not, that's going to determine what happens here in the DCT, those hormones, again, specifically ADH and angiotensin. So this is reabsorption of water. It's also the absorption of sodium and calcium ions. Right? Remember the effects of parathyroid hormone or calcitonin. They're going to play a role in reabsorption of calcium ions. So there's also secretion that happens in the DCT. And by secretion, we really just mean that this filtrate is passed on to go to the ureters, right? To go to the ureters, to go to the urinary bladder. Oh, there's a couple stops on the tour, so to speak, before it gets to the ureters, and we'll look at that later. There's another little guy that's in our nephron, and that is something called the JG complex, or the juxtaglomerular complex. This is a little section. I'm looking to see if you can see it on. You can't see it on 951. Let's see where you can see it. I think you can see it on the map. You can see it on page 952. The JG apparatus or JG complex. It's the same thing. It's in that top right corner of the page. And what this thing is it's closely linked to your afferent arteriole. It's going to control the blood flow into and out of your glomerulus. So it's basically there to monitor the blood flow as it comes into the nephron, and it's going to secrete EPO. Remember that guy, erythropoietin, in charge of increasing red blood cell synthesis. So you have the secretion of EPO. You also have secretion of renin. Renin, angiotensin, ADH, that whole loop that's going to play a role here in the DCT. So this JG complex, again, is really made up of two different types of cells. One's called the macula densa, and one is called JG, or juxtaglomerular cells. Those cells, oops, you can't see macula densa, sorry, macula densa and JG cells. Those cells of the macula densa have the smooth muscle to control the diameter of the afferent arteriole, to increase or decrease what we call glomerular filtration rate in the kidneys, and that's going to have an influence on urine output and urine concentration, and therefore blood volume and blood pressure. So a really cool system that's put into place to help your body maintain homeostasis. So let's talk about some more nitpicky things. This chapter has lots of, well, there's always nitpicky stuff in A&P. Let's take a look at the diagram that's on page 952 and 953. Lots of fun stuff happening. What you see there, and I'm just going to look at A first, and then we'll kind of look through all of them. I'll describe them for you. What you see on A is that renal corpuscle, right, that, or capsule. Corpuscle and capsule are the same thing. Technically, the corpuscle is the glomerulus plus the capsule, and the capsule is just that kidney structure without the capillaries. Just a minor technicality there. But you see the afferent arteriole. You can see that capsule coming around the PCT, a little bit of the ascending and descending loop from the nephron loop, and then the DCT. You can see a little cluster of that JG complex that I was talking about, but not in any detail. You can also see how that DCT, or that distal convoluted tubule, ends in what we call the collecting system, or the collecting ducts. That's again leading towards the finalizing steps of urine formation. In step B, or picture B, you're just kind of zoomed into that corpuscle there, so you can really see closely the special cells that make up the glomerulus, and we'll talk about those in figure C there. You can see that JG complex, the afferent arteriole, how it's closely linked to not only the distal convoluted tubule, but also that JG complex. It's there monitoring, sensing, I call dipsticking, kind of checking the blood, checking the blood pressure, checking the solute concentration, checking the filtrate concentration to make sure that our body's not getting rid of things that it shouldn't be getting rid of, and that it's retaining the things that it needs to. You then have, you can see very closely those specialized cells. Check out those cells of the glomerulus. Doesn't look at all like what you think they would look from figure A, right? In figure A, it just looks like a regular old capillary. But when we get to figure B, we see that the capillary in the of the glomerulus, or the glomerulus as a capillary, is very different. In fact, take a look at letter C, and you can see what we call fenestrations. The cells of the glomerulus are actually arranged. Take a look at them. They kind of like interlock like this, and they have spaces between them to allow for that filtering action. Remember I said the blood slams into that capillary wall, and some of it spurts out and is caught by that catcher's mitt capsule. Well, those little slits called filtration slits are what drives that process. There is kind of a sandwiching membrane there. It's not just these, they're called podocytes. It's not just these podocytes that are there. There is a basement membrane and a capillary epithelium. So there's a couple different layers to get through. So it's not just like any old thing can bust through. But think about this. What happens when your blood pressure is elevated, when you have hypertension, chronic hypertension? You may know someone, maybe yourself, that suffers from chronic hypertension. If you talk to your doctor about hypertension and the long-term effects of having hypertension, untreated hypertension, you will hear several times over and over again, not just issues with your heart, but issues with your kidneys. And think about this sensitive membrane of the glomerulus and why hypertension may be bad for the kidneys. That is such a small membrane, and that blood is slamming into it. As I said, imagine if you have chronic hypertension and it's slamming even harder. What's going to happen is those filtration slits are going to be damaged, and you'll suffer kidney damage and possibly blood in the urine and possibly kidney failure because your kidneys can't filter like they can. So chronic hypertension isn't just bad for your heart and arteries, it's also bad for, as I like to think of them, as intimate capillaries, like within your lungs and your kidneys. So just something to think about. Total side note. The next picture on page 953, what you're looking at here again is the specialized epithelia of the nephron. So I mentioned before in the PCT you have those microvilli for increased reabsorption. That's where the majority of it is coming back into the body, leaving the filtrate, coming back into the paratubular fluid, reabsorbed to the paratubular capillaries, taken back into circulation. Within the nephron loop, both the ascending and descending, what we're talking about here is just a thin layer of simple squamous that's to allow for quick, easy exchange through that countercurrent multiplier, where as the solutes are leaving one side, and you can almost imagine how that might play out there. Solutes are leaving one side, that's going to pull the water from the other side, uber concentrating that filtrate. Then in the distal convoluted tubule, some of them have microvilli, and again, it all kind of depends on what the hormones are saying to have. Then within the collecting duct, again, a little bit of my microvilli. The collecting duct, even though it's technically not part of the nephron, the solute concentration can still change in the collecting duct. The filtrate composition can still change within the collecting duct. It's not over till it's over, as I like to say. That collecting duct still has some variables in it, still has some cells that can do some reabsorbing. Nothing's being secreted back into the filtrate, but if for some reason your body all of a sudden decides that it doesn't have enough water, more of it can be reabsorbed even from the collecting ducts. 954, we already talked about blood flow, and I think we'll stop here for this video because we're approaching 15 minutes.